Hello everyone, and thank you for attending today's webinar, Bioanalytical Project Management, a critical function adapted to the study types of your drug development program. I'm Martha Powell, Editor of Infectious Diseases Hub at Future Science Group, and I'll be your host for today's event. Before I introduce you to our speaker, I'd like to quickly cover a few housekeeping items. Firstly, all the widgets are resizable and movable, so feel free to move them around to get the most out of your desktop space. You can expand your slide area or maximize it to full screen by clicking the arrows in the top right-hand corner. Please let us know your thoughts on today's webinar by tweeting us at Bioanalysis Zone and using the hashtag BZWebinars, or simply share today's webinar with your friends and colleagues using the share widget on your screen. Some networks can cause slides to advance more slowly than others, so logging off your VPN is recommended. If your slides are behind, pushing F5 on your keyboard will refresh the page. You can find additional answers to some common technical issues located in the Help widget at the bottom of your screen. Finally, an on-demand version of today's webinar will be available approximately 1 hour after the event and can be accessed using the same audience link that was sent to you earlier. So we're to have Anahita Keyhani from Altasciences presenting for us today. Anahita joined Altasciences in May 2015 and has over 20 years of CRO experience in regulated bioanalysis for preclinical and clinical development. As Senior Director of Scientific Operations, Mass Spectrometry, Dr. Keyhani leads a team of over 40 scientists dedicated to method development and innovator-regulated bioanalysis, both preclinical and clinical. Prior to her current role, she was responsible for method validations and bioanalytical sample analysis projects for all phases of drug development. We invite the audience to submit any questions you may have for our presenter throughout the webinar using the Q&A widget to the right-hand side of your screen. We'll pose your questions to Anahita in a live Q&A session after the presentation and then follow up any outstanding questions offline. Thank you again for joining us today, everybody, and I'll now hand over to Anahita to start today's presentation.
Good morning, and thank you, Martha, for the introduction. Today, I will discuss bioanalytical project management. I will review selected critical control points and the framework and information transfer for successful progression of the bioanalysis within the drug development program. Given my background and experience, the presentation has a CRO perspective and examples are from mass spec. Just a little information about Altasciences. We are a mid-sized full-service contract research organization with over 25 years of experience in drug development. We have 2 clinical sites in North America. One is in the U.S. in Kansas City, and the other is in Montreal, Canada. We also have a preclinical test facility in Seattle, Washington, acquired in the summer of 2018 to help us achieve our goal of providing an integrated research service solution from lead candidate selection to proof of concept. We have 2 laboratories, one at the Seattle preclinical site and one near our Montreal clinic, with 25,000 square feet of laboratory space that supports both small and large molecule analysis. Our laboratories offer comprehensive bioanalysis services for studies at our clinical sites and test facility, as well as offering bioanalysis as a standalone service. As an overview, keeping in mind that successful bioanalytical project progression is based on teasing out the details that are unique to each study, this presentation will include some thoughts on the development pathway, considerations for materials, method development, assignments, and framework, and also the implementation of new technologies and some concluding thoughts. Bioanalysis is a significant component of the drug development pathway. As technologies and materials have evolved and improved, the bioanalytical options have increased. For this presentation, we will focus on the bioanalytical support of new molecular entities. The considerations for bioanalytical project management depend on study type and scope. As development progresses, information is compiled on safety and efficacy. Bioanalytical results from one study can be the driver for additional studies. Along the development pathway, gaps in data and knowledge often become evident and need to be addressed. Following initial success in the clinic, non-clinical studies and clinical can be ongoing simultaneously. As bioanalytical scientists, our objective is to generate accurate and precise data based on an understanding of the defined variation of our methods and uncertainty tolerance accepted on the data. The integrity of the sample has to be maintained from collection till analysis. The schematic is an example of various stakeholders that may share information to ensure the alignment of sample collection and bioanalysis. It is important for information to be transferred at the right time with the appropriate stakeholder. Scientific and statistical evaluation of the results from a single study or over several studies allows meaningful decisions on safety and efficacy. The studies and/or analysis can also address emergent science and risk-benefit of addressing gaps in knowledge and data at a certain time in the development pathway. The next section focuses on general considerations for materials as they relate to timelines and schedules. To initiate a project or a series of projects and lock in timelines and deliverables, the availability and quantity of reference standards, internal standards, critical reagents, and consumables needs to be determined. While laboratories maintain an inventory of materials routinely used in their workflow, procurement timelines need to be considered and managed for specialty materials. The review of the documentation, such as certificates of analysis, should occur as the materials become available. This provides time to discuss and address any questions and evaluate based on the project or projects for which the material will be used. As a very simple example, if the CLV is not suitably clear to determine potency calculations for stock solution preparation, discussion and agreement may be required. Some questions to ask are the materials provided for method development or non-GLP sample analysis suitable for validation or regulated sample analysis? At the point of transition for studies in compliance with regulations, an assessment should be made. If critical reagent quantities allow, reserve an inventory of each lot for at least 2 to 3 analysis batches to provide opportunity for bridging if required. Reference materials may be modified along the development pathway, for example, new salt forms. Potency adjustments are simple. Existing methods and solvent used for stock preparation should also be evaluated due to the modification. Success or challenges in the development pathway often result in additional studies. With the expansion of the program, the quantity of materials available is a key consideration. In case of specialty materials, there should be proactive planning for long-term studies or several studies within the program, and procurement timelines need to be considered. The method is the cornerstone of bioanalysis. The statement "begin with the end in mind," for me, as a bioanalytical scientist, can be broken down to mean the bioanalytical sample collection, processing and storage, the bioanalysis itself, and finally the end use of the data. I wanted to take the opportunity to discuss method terminology. In addition to traditional TK-PK sample analysis, bioanalysis now often includes tissues, biomarkers, immunogenicity, among other possibilities. Bioanalytical support at all stages may now be shared by the innovator company and multiple CROs. Niche bioanalytical capabilities requiring specific instrumentation and scientific talent are supported by specific laboratories. With the ascent of the CROs as partners in development, method categorization, such as screening, discovery, qualified, validated, fit for purpose, is now in discussion. When bioanalysis was conducted at the innovator company, any limitations or gaps were managed as the program progressed. And in my opinion, such precision in method status was probably not needed for every study. The method should provide meaningful and scientifically relevant data for decision-making. When a validated method is presented as an option, the thought that comes to mind is trust and verify. A method is developed and/or validated at a specific time in the development program to support a specific study. With every new study, there needs to be a verification that the study samples are in alignment with the method. Basic verifications include the suitability of the calibration range, the available sample volume, any impact of co-administered compounds. For drug-drug interaction studies, is there any metabolism that can be impacted that can impact the performance of the methods? Matrices from patient populations. At what point do they need to be assessed? Or do they need to be assessed? In all cases, a theoretical evaluation of a method vis-a-vis the study is needed, and additional experimental work in development and/or validation may be required. For de novo method development, structures and any available information should be shared to ensure the best development path. Assumptions on the transferability of methods should be avoided. Providing available methods and development and validation reports for review is recommended. If development for a non-clinical study is needed and clinical studies have already been initiated or completed, providing both clinical and non-clinical methods for review is suggested, so that the totality of the scientific information is available before transferring and/or developing a new method. The FDA 2018 and ICH-M10 do discuss equivalency of temperature for storage between -20 and -80 C. In my opinion, assessment of both temperatures during method validation is not a significant amount of additional work and is much more efficient than coming back for these assessments later, when samples may not have been stored at the appropriate condition, especially for larger multi-site studies. Procedures and preservatives selected for sample collection at a test facility or phase 1 clinic may not be appropriate for a hospital, doctor's office, or certain geographic locations. For sample collections requiring preservatives, the workflow and logistics should be considered. Including validation assessments with and without preservative for future impact assessments is suggested. There should always be communication between stakeholders to align scientific and logistical requirements. For complex or novel sample collection procedures, providing schematics, video training clips, or even on-site training is helpful. In this slide, there is an example. A recent study required the collection of samples for 3 analytes. The analytes could not be combined in a single method, and to achieve timelines, method development of the analytes was ongoing concurrently. 1 analyte required preservative, and the other 2 were simple collections in K2-EDTA tubes. The study was multi-site with activities occurring in doctors' offices. Considering the workflows and technical competencies at the site of sample collection, it was decided to simplify the sample collection kits and harmonize the collection procedures. While the preservative was not scientifically necessary for all 3 analytes, simplification of the procedures at the site of collection ensured minimal collection deviations and reduced complexity for the staff manipulating the samples. In this section, we will address assignments and framework for bioanalytical support of non-clinical and clinical studies. I'd like to discuss the organization and assignments in GLP studies. For non-clinical studies, the OECD GLP multi-site guidance defines the roles of principal investigators for phases of the study conducted at sites organizationally or geographically distinct from the test facility assigned study director. For scientists at the test facility, the assignment is usually adjusted to contributing scientist or project scientist. The study director is responsible for the GLP study. The GLPs and multi-site guidance provide a framework to ensure appropriate study planning, communication, and study control. Bioanalytical principal investigators are assigned for the bioanalytical phase, and there could be several assigned, depending on the analysis and/or test sites identified. The study plan or protocol is the driving document for studies with an assigned study director. The study director is responsible to ensure the protocol and all amendments are available to the assigned principal investigators in a timely manner. Scientists assigned to a particular phase can also request amendments to the protocols for the details related to their phase and to facilitate their work. For initial non-clinical studies, method development is often initiated as the study director is developing the protocol, and it is best practice to assign the principal investigators or study scientists at this time. When a scientist is assigned as the bioanalytical principal investigator for validation and/or sample analysis is different from the individual or team responsible for method development, it is best practice to include the bioanalytical principal investigator in communications regarding method development, such as emails, conference calls, and internal meetings. Having the assigned bioanalytical principal investigator in contact with both the method development scientist and the study director ensures the appropriate and efficient transfer of information for inclusion in the protocol. Principal investigators are responsible to provide all deviations of their assigned phase to the study director for impact assessment. Study directors should also provide deviations to sample collection, process, and storage to the PIs responsible for bioanalysis and statistical analysis at the appropriate time. In larger companies, the sponsor representative for bioanalysis is often a different individual than the sponsor representative on the protocol. This results in requests for bioplans and laboratory phase plans for non-clinical sample analysis. This should be discouraged, especially for GLP studies. If the sponsor contact for a specific phase is different than the main sponsor representative for the overall study, their names and contact information can simply be included in the protocol. When the timing of non-GLP dose range finding studies allows for analyzing TK samples with a validation-ready or qualified method, proceeding with the analysis at this stage is recommended. The sample results will provide opportunity for unexpected method adjustments, such as calibration range or chromatography adjustments, to be implemented prior to formal validation, and the sample analysis for the IND-enabling studies. If there is a validated method for a specific species and strain, each non-clinical study needs to be evaluated to ensure that the validated method is appropriate. The routes of administration, sample volume, dose levels, formulation adjustments need to be assessed against the validated method. Changes that can lead to increases in sample concentrations, as an example, using a method initially validated for an oral gavage study to analyze samples now being produced from an IV bolus, can usually be addressed with sample pre-dilution schemes to reduce sample repeats. Study designs and dose levels and test formulations that now require a lower limit of quantitation to produce meaningful data may need additional time and resource allocation for method development and/or validation. Scientific evaluation of transferring methods for validation between species and strains is required, and assumptions on a simple transfer between matrices should be avoided. Non-clinical protocols often include proposed dates for animal arrival, dosing dates, draft reports, and final reports. Proposed dates for different phase reports are included, or it is indicated that the dates will be added by amendment. A critical step in the timeline is often the transfer of bioanalytical data to another PI for statistical analysis. This transfer date must be determined and agreed upon to ensure the project progression timeline. Having direct communication between the assigned principal investigators or project scientist and the study director on this critical handoff of data is valuable, and the impact of date adjustments in providing the bioanalytical data needs to be monitored closely. In earlier development, there is potential for unforeseen issues impacting the method development, validation, and even sample analysis progression. The bioanalytical scientist being aware of all the linked projects, development, validation, and sample analysis allows for prompt evaluation of any challenges and the impact on project progression. For example, method development scientists, the bioanalytical PI, the study director, the statistical analysis PI, and the appropriate sponsor representatives should be included on the communications. So we will now move on to clinical studies and bioanalysis. For clinical studies, in addition to the clinical project manager and many other contributors, bioanalysis requires individuals with scientific, technical, and project management competencies. There is no formal framework for bioanalytical phase management. The assigned role and responsibilities of the bioanalytical project scientist may be based on the clinical study type and organizational structure. So assumptions should not be made when working with a new organization, and it is suggested to obtain clarity on roles and responsibilities. Clinical protocols are sparse regarding bioanalysis details. The bioanalytical methods in alignment with the protocol need to be determined. The information on sample collection, processing, and storage and shipment is often included in the laboratory manual or similar document. The bioanalysis details are included in laboratory phase plans or bioplans, which include the assigned scientist, the details of the bioanalysis, the methods and the validation references, sample storage at the laboratory, and references to reporting and archiving. For successful project progression, the document should be shared for review between the different stakeholders prior to finalization and at the appropriate time. For first-in-human studies, the bioanalytical sample schedule is based on the dosing and safety meetings and requires some discussion. There needs to be precision on sample transfer, shipment, and sample analysis, the resource allocation such as staff and instruments, and provisions for data release. Discussion prior to the study start on handling of samples above the limit of quantitation is beneficial. Can extrapolated values be provided and repeats with dilution conducted in another batch, or are repeats with dilution required for the safety meeting? Is there an acceptable uncertainty on a batch for release of some sample results? This is a crucial conversation for analysis that have long injection times for mass spec or long workflows, such as immunoaffinity mass spec when extractions can have a 2-day workflow. Having these discussions will allow appropriate scheduling for dosing, safety meetings, and sample analysis. Often first-in-human studies have urine collection, and requests for sample analyses are dependent on the review of the plasma serum data from initial cohorts. However, to obtain meaningful data in the event that urine sample analysis is required, initial development to determine sample collection procedures, such as evaluation of nonspecific binding and collection containers, are suggested, even if complete method development and/or validation is on hold. Validated, rugged, and robust methods on specific platforms should be available prior to the initiation of the later stage clinical trials. In my experience, challenges to the bioanalysis do arise as a result of consumables and critical reagents. As an example, additional peaks in the chromatogram were traced to a new mat lot used to seal the injection plate. This was finally attributed to a mat manufacturing process change after some investigation. In this case, minor method development and validation were needed for adjustments to chromatography. In another program, the critical reagent provided by a qualified supplier could no longer be produced with the same performance as previous lots. The method had to be completely reworked and a new validation conducted. For studies with a long duration and multi-site studies, sample shipment and analysis schedules should be considered in terms of the validated long-term matrix stability. Extending the stability as the program progresses to reduce risk is recommended and not leave this assessment for study end. As programs expand with different types of clinical studies, the methods and validations need to be evaluated for suitability. As an example, the impact of co-administered compounds. For multi-site and long-term clinical studies, logistics of analysis becomes a critical component in ensuring successful bioanalytical project progression. Discussions on the points in the table, such as the number of samples expected, the number of samples per shipment, when is the clinical study starting, the first subject, first visit, the expected end of the study, the last visit of the last subject. What are the frequency of shipments? Will it be sample shipments be weekly, monthly, quarterly? What are the turnaround times needed on the samples? When is the data transfer agreement going to be put in place? And what is the frequency of data transfer? Who is blinded and who is not on the bioanalysis? Setting an initial baseline for allocation of resources and planning is very beneficial. Of course, there is fluidity, and once the base is set, it can be adjusted in real time. Speaking from a perspective of a CRO, it is better to plan tentatively and have slippage out or cancellation for planned sample analysis rather than triage it in unexpectedly. Of course, urgent bioanalysis due to adverse events or other reasons always has priority, and my comments are more for routine analysis. For these clinical studies, there is the data flow, the data cleaning, discrepancy resolution to manage between multiple stakeholders. There can be missing samples or extra samples at the laboratory or in the database, labeling and documentation issues, and shipment oversights. While there are many stakeholders, many of the queries and issues make it to the responsible bioanalytical scientist. To facilitate project progression at the initiation of the project or program, the roles and responsibilities of the key stakeholders should be defined in addition to communication and escalation channels. The collaboration of the bioanalytical scientists is often with sponsor representatives and data management teams, and even other stakeholders. Schedules need to be managed for test files, production files, data transfers, soft locks, and hard locks of the database. Clarity on planned milestones allows for efficient project progression. In addition to the sample analysis, efforts should be made to set tentative schedules for QC data, QA data, and reporting timelines, and even reporting formats. So last but not least, given the moment in time with the merging of infotech and biotech, advancements such as software and computing power, precision manufacturing, and the variety and capabilities of instrumentation provides us with the opportunity to implement new technologies for innovative bioanalytical solutions. Our approach for transitioning a new technology to routine use has been to review the bioanalytical method validation guidances, the scientific information, and the end use of the data to establish a method development plan. We evaluate the impact of the new technology on our standard logistics and workflows and determine the adjustments that need to be made. We review the content of our general SOPs and look at our method SOP templates to identify and address the impact of the new technology. Stakeholders need to collaborate and manage the progression from development to routine use in the laboratory. The validation plan is developed with the scientifically relevant experimental assessments and acceptance criteria. A skeleton SOP for the novel technology is put in place, and as the technique is applied to several validation and analyses, the SOP is expanded upon to standardize the required assessments and workflows. As predicting every eventuality is unlikely, the framework of the GLPs is a good approach. This ensures any deviations, logistical, and workflow challenges are captured, discussed, and addressed in the study file and report. As an example, I will discuss the implementation of Mitra, volumetric absorptive microsampling, in our laboratory. The potential of the device in supporting pediatric studies, home sampling for adherence, and therapeutic drug monitoring and enriching data sets was recognized. Our initial focus was for small molecules. We evaluated against a plasma method validation in terms of documentation, workflows, and experiments. For clinical validation, precision and accuracy was also assessed for QCs extracted with blood hematocrit levels at minimum and max levels relevant to the study. While stabilities at frozen temperature were not required, other stability assessments were identified. As an example, for methods requiring plasma or serum, whole blood stability is usually assessed for 2 hours at room temperature. For preparation of a pool of Mitra standards in QCs, extending whole blood to 8 hours at room temperature simplified the workflow. Procedures were adjusted if the stability of the analyte in whole blood did not allow for this duration. In addition to long-term stability on the Mitra microsample at ambient temperature, the potential storage conditions and oversights needed to be considered based on possible sample collections. An example is the omission of desiccant in the storage container, which we saw multiple times. Consideration should be given if the VAMs are being collected in a specific geographic area with elevated temperatures and high humidity. Subject samples are often only 2 Mitra and therefore allow for 2 extraction attempts. The assessments of stability in the primary extraction plate, in addition to the injection plate, were included in the validation. This allowed that ULQ samples could be diluted, since we would extract multiple internal standards in the batch and use those to dilute the samples and repeat them. As with every new technology, experience and new scientific information requires us to often review SOPs and workflows to determine if adjustments are needed. So some concluding thoughts. Effective and efficient bioanalytical project progression requires proactive collaboration between internal and external stakeholders. The lines of communication and responsibilities need to be established. For GLP studies, efforts should be made to capture the information in the protocol. For clinical programs, the RACI framework is useful to identify who is responsible, who's accountable, who's consulted and informed for the bioanalytical component of the study or program. Critical control points for bioanalysis management are unique to each stage of development and study type. The implementation of innovative bioanalytical technologies are best served with adhering to a GLP framework. So with that, I thank you very much for your attention.
Thank you for that very insightful presentation. I hope our audience found it of great interest and value. If you do have any questions for Anahita, then please continue to submit them using the Q&A tool in the right-hand side of your screen. We've received a number of questions so far, so we'll address some of these now live on air. So our first question, my organisation requests a bioplan distinct from the tox protocol for the bioanalytical phase of GLP studies. What are your thoughts?
As indicated in the presentation, this is not the best option for GLP studies. Study directors can easily make amendments to their study plans, protocols to include additional information, for example, who's the bioanalytical contact at the sponsor site, or where samples have to be shipped after analysis, maybe back to the sponsor. So I do not suggest that for GLP studies a bioplan is used.
Great, thank you. Our next question. The Mitra microsampling is very interesting. Have you experienced any issues with ISR from dried blood matrix? And what sorts of long-term data have you generated in your validated methods?
Well, till now we validated 9 methods, and in one of them we have up to 2 years stability. For the methods that we have done samples and have done ISR, we haven't had any issues. This could be part of our technique, which is called impact-assisted extraction, which you can read about in posters, publications, there's information in the public domain.
Great, thank you. Can you elaborate on when you might use a screening versus a qualified method in the early stages of a drug program?
Okay, so I will comment for preclinical work. So in non-clinical development, a screening method is used for analysis with an understanding that the uncertainty on the results are due to the method not being completely interrogated. A qualified assay should be used to generate data that will be used for design of a GLP study. So for example, as I mentioned in my presentation, dose range finding studies are usually non-GLP. And the results then have to be used for setting dose levels in a GLP study. So even if the method is not validated, the results will be used for definitive dose selection. So that's sort of the difference I see between screening and qualified assays in non-clinical work.
That's great, thank you. Thank you very much, Anahita, for answering our audience questions today. Before we wrap up, do you have any last comments you'd like to add?
No, just thank you.
Great, thank you. I'd like to once again thank our wonderful presentation from Anahita, as well as you, our listeners, for your time and for your questions. If you'd like to let us know your thoughts on today's webinar, then please feel free to complete our feedback survey in the resource widget or tweet us at Bioanalysis Zone. And don't forget to visit us at www.bioanalysisZone.com for more webinars. Thank you again for attending, and goodbye.